Okay, hello everybody. My name's Keir, and you already heard more than you would like to hear about me. But I started on my group at the Biohub 3 years ago in pursuit of holistic pictures of infection, from the moment a virus invades a host cell and onwards through all paths that lead to organismal health or disease. And we heard some good news, or at least interesting news, from Artem, and that's that we're swimming in a vast sea of viruses. And with sequencing, we've gotten pretty good at finding new viruses just about anywhere we care to look, as he showed us, whether that's a human, a bat, or a whale, or a penguin swab. But where we've struggled a bit more is in making functional guesses about these new sequences we're discovering. Ultimately, we have to do some kind of experiments to figure out what these sequences can do. And that holds even for new variants of viruses that we know quite a lot about in other contexts. Now, one approach where we can gain some traction here is by carrying out virus discovery in an experimentally tractable host context, where we have a built-in set of tools we can use to learn about, study any new viruses we might happen to discover. And I'm going to make the case that zebrafish is a really nice organism to carry out this kind of approach with. But just to distill the key points down, it's unique amongst the vertebrate research organisms in being amenable to whole organism profiling, but at single cell resolution. So when I was starting this program, no viruses of zebrafish were known. And so when I was a postdoc with Nels, I built these fish that glow green in response to interferon signaling. And I was surprised to find that it was exceptionally easy and somewhat unexpected to find glowing green fish before I had done anything outside of the laboratory. So here they are swimming around in their petri dishes, and they were seemingly spontaneously turning green. And then through sequencing these green fish, pulled out a new member of the Corniviridae family, which looked just like other members of these vertebrate infecting viruses and the key features of these viruses. And it turned out, as you heard, it wasn't just in these laboratory fish swimming around in Utah. I can look in publicly available data, and this is a very minimal search across all the possible public data, but could find evidence for this virus in research laboratories around the world and other people studying zebrafish. Right, so this was a prevalent, undetected virus previously to this, and this opened up the immediate question of, is this the only virus that's swimming around with these zebrafish, or can we actually find other examples and start exploring the vast variety of ways that viruses interact with their host with this system where we have all kinds of nice handy tools? So to address that question, Itria, Eric Duo, and essentially the whole computational biology team at the Biohub has constructed this incredible pipeline, which I won't get into the details here, but please talk to Eric at the poster session to ask him more about how this actually works. But the core features of it are that it takes the great variety and ways that people have produced sequencing information and parses the technical from the biological signal in that information to start. You could apply this to any organism, but in this case, we're focusing on zebrafish, so we've looked at more than 70,000 RNA sequencing data sets that are currently housed in the Sequence Read Archive. And with that information, we do two different things. One, we use the signal coming from zebrafish to infer things about the gene expression in that host. And then with everything that's not zebrafish, we do essentially what Artem was describing and assemble it and try to infer some kind of taxonomic label to understand what that virus might be. OK, so of course, there's much more than just viruses in there, but that's what I'll focus on today. And the big picture here is then to move from this in silico detection, discovery, and hypothesis building into experiments that we can go back and use the real strengths for zebrafish to figure out in more complex situations. OK, so just starting with the viruses, Eric has come up with this really nice way to visualize all the sequences that we pulled out of zebrafish sequencing data that we've inferred are some kind of virus. So each tip here is a unique virus taxa that we've inferred is where the sequence is coming from. And I'll zoom around different parts of this tree just to give you some examples of what we're seeing here. So a huge chunk of this down at the bottom are bacteriophage. These are not, of course, directly infecting zebrafish, but perhaps interacting with the microbiome that's interacting with zebrafish and driving their biology in different interesting ways. But that's a lot of it we won't consider today. we do see the first virus that I discovered and described, the zebrafish picornavirus, but also other relatives we hadn't seen before in the Picornaviridae family. We see some maybe unexpected, depends, examples of viruses, including SARS-CoV-2, and this is not the source of the pandemic that Artem was looking for, I don't think. In fact, we know it wasn't. This only shows up post-2020, and this is a clear case of sneezing in the samples that is essentially coming from library preparation, generating sequencing data. So this is just an example of things that are in there that are not, of course, directly interesting for understanding how viruses are fighting infection, or how zebrafish are fighting virus infections, but are something to contend with in the data. Then I'll give you just one example of something that we think is interesting and is significant for being a legitimate infection of zebrafish. And this is an example where the closest relative to the sequences we found to this virus looked like they were coming from some other kind of fish. And in this case, is an influenza virus. And I'll just zoom in on this virus in a little greater detail to give you some examples of why it's so striking. So a few things stand out about it. Here I'm color-coding the 8 different segments from this influenza-like virus that we have detected and discovered. And the coloring here, going up to blue down to yellow, is the amino acid identity of that, the coding region of each of these segments to, in this case, human influenza B virus. And you can see in the polymerase segments here are dark blue, and that means they're highly similar to human influenza B polymerase proteins. And this is up to 90% amino acid identity. Whereas everything on the virus envelope, in this case, is extremely distantly related, much more closely related to weird fish things. And the strangest example is even M1 and M2, which are encoded on the same segment, but in different open reading frames. M1, which is found on the inside of the envelope, just on the inside of the envelope, is closely related to influenza B, whereas M2, which spans that envelope, is highly diverged from the human virus example. So just to illustrate this phylogenetically, if we use, for example, PB1, one of the replication proteins from this virus, you can see that this virus we found in zebrafish data clusters closely with the human influenza B segments, whereas if we look at, for example, NA, it falls as a relative to this Wuhan spiny eel influenza virus. So what exactly this means is still unclear, but also, and everyone may be familiar, but is particularly weird because there's no known non-human reservoir for influenza B. So it's pretty weird that we would find something in zebrafish in this case as being the closest relative to this human infecting virus. But although this might look exotic and strange on the surface, one thing it illustrates more generally is that the viruses we're finding in zebrafish all have counterparts in the human world that that make us confident that by studying infection in this context, we'll learn something about virology and immunology that spans across organisms. Okay. So now turning away from just the virus sequence information alone into inferring other things about the interaction between its hosts, we've turned to the host transcriptome data as another layer of information we can use to make us confident that this is actually an infection happening in zebrafish. So was there a response to that virus sequence? And to start with a simple example, Duo on the comp bio team asks, essentially, what if all these data sets were coming from our green fish, essentially? What if we just look at ISG15 expression across all sequencing data sets and look for the elevated cases of ISG expression in those data? So here, plotting the distribution of, in this case in the left panel, all virus negative runs across the zebrafish sequence read archive, their distribution of ISG15 expression on the x-axis, compared to all runs containing the zebrafish picornavirus, which we know, at least in some contexts, leads to elevated ISG15 expression. We see that shift to the right, suggesting that there's this general trend in that direction. And then just as one other example out of all the other viruses we've looked at or discovered so far, the flu virus also seems to hold that pattern. So the data sets that contain these virus sequences tend to be shifted into some antiviral immune response. This is a very simple scenario. We can also, of course, leverage the entire transcriptomes in those data sets to get a more nuanced picture of what's going on. And Duo started to do that by taking some shallow modeling approaches, where we're now considering the entire transcriptomes and building a classifier to try to tell us, does it think that this particular transcriptome or data set is associated with some virus infection response? And we can, of course, build on this and leverage more recent, deeper learning methods to interrogate not just, is this particular sample infected by something, but do we have unique responses to different kinds of insults here that allow us to infer things about the general immunology at work? And so I think Artem really elegantly articulated the power of using these kinds of data to propose hypotheses that would be interesting to follow up on with further experimentation. And that's absolutely what we envision doing with this kind of information. So now we want to move beyond just the in silico predictions of what's going on into actually visualizing in a dynamic way how an organism, a vertebrate, can resolve all these different varieties of insults to their systems. And I'll start by just talking about our work with the zebrafish picornavirus. So to step back a little bit into the broad categorization of what happens when fish are infected by this virus, even in these green fish here, we've observed that fish get infected, they mount an immune response, and then they clear the infections. And then presumably over their lifetimes, they get infected, reinfected, and go through this process again and again. But how do they do this? What's going on here? This is what we want to figure out, what we mean by immune response with much greater resolution that zebrafish experimentation affords. So what are the kinds of things we'd like to know? We'd like to know where the viruses are in this picture. You're looking again here, the green is a reflection of response to interferon signaling, not necessarily where the virus is, which cells it's in. We'd like to know where does the response to that virus start and extend to and get resolved by. And we'd like to know what cells are involved in coordinating that resolution of this kind of insult. And how does all this go wrong occasionally? So I'll just start with an example of looking at where the viruses are. This is something that Jared and the group has devised a really nice reporter for, in this case, reading out the activity of the virus. So here what he's relying on is he's anchoring a transcription factor to the membrane that's tethered to the membrane with a sequence containing the cleavage site. A cleavage site that he defined is used in the virus genome itself to be cleaved as the polyprotein is processed, so that when zebrafish picornavirus is in the cytoplasm and gets translated, its protease will cleave this transcription factor from the membrane and go on to activate gene expression in those host cells. And just zooming in on one example here, this is early in his production of this line, but seeing some really encouraging signs. We know from other data that the virus is localized to the gut. And here we're starting to see gut cells line up in the anterior part of the zebrafish gut here, telling us for the first time where this virus is in living animals. Jennifer in the group is then asking about what happens next. So what's the response to these infections? Can we look at this in a dynamic way? And so on the video on the top, she's showing you the sort of pre-infection and post-infection wave of activation of interferon signaling, where you're seeing the same IHG15 GFP fish get progressively greener over time. And she can then take snapshots of this process in fixed animals to ask more nuanced questions about not just where are the fish turning green, but which cells are actually infected, which cells are responding to infection. This gets at the heterogeneity of responses that have been a really great theme of today's meeting here, where if you zoom in, we can look at individual cells now. These are stained to identify the transcripts of the virus in orange, or the production of interferon in magenta. And we're going far beyond the textbook examples here, where it's not a one-to-one relationship of if you're infected, you produce interferon or something else. There's a lot more going on here, and this is something we would like to resolve with dynamic information. Okay, so, and then in addition to the immune signals and how this coordinates response, the last piece of this we'd like to get at is the immune cells and how they're involved in mediating this effective clearance of infection and other insults. And to get at capturing the immune system in its entirety, we've generated transgenic fish where every immune cell produces mCherry protein, but no other cells in the fish do. And when Deepika teamed up with Sheng and our colleague Loic Royer's group, who built a one-of-a-kind microscope that produces exceptional depths of imaging volume, and watched these fish over time. This is not in real time. This is sped up, so what you're watching is essentially 24 hours of real time. And what you can see is an incredibly dynamic process of these cells cruising around. And what these cells are, Jared sorted out over time and identified essentially every counterpart of cell type that we know about in the human immune system is something that we see crawling around in these zebrafish, telling us that by studying infection and other kinds of inflammatory insults in this context, we could learn about a human's immune system that looks a lot like ours. And we want to do more than just, of course, observe what's going on here. We want to be able to quantify and describe in greater detail. And to do that, Deepika has been building custom tools to be able to segment the individual cells here. And she's working with Jordao and Loic's team to push this world-class cell tracking approach that he's devised so to follow all these cells over time. And what Deepika's done in this particular video is color-coded all the tracks of where those cells are going based on their average velocity across the entire data set. And so you can immediately get a sense for differences across the system. And we think, in addition to getting at here just the movement of cells, Deepika is pulling out information about the morphologies, their shapes, and how they change over time. And this is just one example of a cell now that's crawling over to Jordao. And using that information together with the traditional modes of profiling the molecules that make up these cells and responses they make to these insults in gene space, we think we can make more holistic measurements of what we really mean when we're talking about an individual cell type or state and how that changes in dynamic ways. And so we're building all of these tools up to capture the systems level measurements and understanding of the immune system organization and function in the context of virus infection and many other inflammatory insults. So I'd like to thank my amazing team and colleagues at the Biohub, and I'm looking forward to talking to everybody more.